place at the basolateral surfaces by means of sodium-potassium ATPase pump shown. Now, there is not just one pump which is responsible for primary active transport. There are a number of such pumps present in various parts of the tubules. They can be hydrogen ATPase, hydrogen potassium ATPase, or calcium ATPase. So all these, they derive energy from ATP and they subsequently cause movement of other ions because of their activity. The basolateral sites have extensive sodium-potassium ATPase. It maintains low sodium concentration inside the cell. It also maintains a net negative charge of -70 millivolts and thus favors passive diffusion of sodium from lumen into the cell. Then there are certain sodium carrier proteins also present on the luminal surface, and due to these carrier proteins, facilitated diffusion of sodium also occurs. And when there is facilitated diffusion of sodium, along with it, through these carrier proteins, certain other substances are also transported, and they constitute secondary active transport, like glucose and amino acids. This will be clear from this figure. So what we see here, again, this is the luminal side shown here, and this is the basolateral surface of the cell. We can see that on the basolateral surface, we have already discussed the primary active transport taking place, that is sodium-potassium pump is functioning here. And as I told you, the gradient of sodium will be directed towards the interstitial tissue because sodium pump is forcing free sodium ions towards the interstitial space. And due to this gradient, sodium will enter by various means. It will enter passively. It will enter by facilitated diffusion. This is the channel. This is the carrier protein. So this carrier protein will drive in sodium and along with it, glucose also enters. So this movement of either glucose or amino acids shown here is secondary active means of transport. Okay, so these are the substances which are traveling inside, or we can say they are reabsorbed by means of secondary active transport, right? And this is also an example of co-transport because they are being transported along with sodium. Another substance which is secreted here in the tubular lumen is the hydrogen ions. Again, this is an empty port. When sodium is moving inside, due to its gradient, hydrogen at the same time is secreted out. This is also an example of secondary active transport which is taking place. So we can see that hydrogen is secreted by this particular mechanism due to sodium, and amino acids and glucose are transported inside the cells by secondary active transport. Now there is a very important term here known as tubular transport maximum. Many of the absorptive and secretory mechanism depends on carrier proteins, as we have just now discussed. In case of sodium, glucose, amino acids, so these substances, they need a carrier protein to be transported inside. So the maximum amount of a substance that can be transported depends on the number of carrier molecules available. And there's a limit to the amounts of the carrier molecules, availability of the carrier molecules. So when all the carrier sites are occupied, then we say that that particular carrier is saturated. So most substances which are transported by carriers, they display a transport maximum indicated by Tm. That means there is a maximum limit to which this substance can be transported. I'll repeat what is transport maximum. Many substances which are transported due to by means of carriers, they have a limit to be transported because carrier can carry a particular amount of substances only. There is a limit to the number of carriers present on the membrane. So when all carrier sites are saturated, no more substances can be transported. So there is a maximum limit to the transport of substances which follow this means. So this is known as Tm for a particular substance, and here Tm for glucose is represented by Tmg, and its value is 375 milligram per minute. That means that this much amount it can be transported per minute. Beyond this amount, it cannot be transported in the tubules. Now this graph represents three processes. First of all, see the x-axis. What we see here is the plasma glucose concentration. I have taken the example of glucose because that is most important in the function of the body. So plasma glucose concentration is depicted on the x-axis, you can see. And on the y-axis, first of all, we'll consider the red graph, which is showing glucose filtered load. Okay. And as you can see, this graph is just a straight line, or we can see it is directly proportional line, which shows that more and more plasma glucose concentration is, more and more glucose will be filtered from the glomerulus. Okay, so the filtered load of glucose will be directly proportional to the plasma glucose concentration. This was the red part, which is indicating the filtered load. Coming to the blue line, this is indicating the reabsorption of glucose. So this indicating, the blue line is indicating the reabsorption of glucose. Again, when the plasma concentration of glucose keeps on increasing, the filtered load is also increasing, and the reabsorptive capacity is also increasing. But as I told you, since it is a carrier-mediated transport, it has a limit. So at a particular limit, say 325, at that particular limit, it can no more be reabsorbed. So you can see that this curve has flattened. Okay, so reabsorption of glucose will not occur beyond a maximum limit, and this is known as TMG, transport maximum of glucose, because all the carriers has been utilized now, and so much load cannot be handled by the tubules. So that is the transport maximum of glucose. Now the green line, the green graph, is indicating the excretion of glucose. So once a threshold is reached, that is a level is reached in the plasma. Here it is indicated by 200 mg/%. So when the amount of glucose increases beyond this, the glucose will appear in urine. So that is indicating the excretion of glucose in urine beyond this particular limit. All right. Now you can see that here, this curve is a curved curve. It is not the straight curve, as I'll show you in the next figure. Here you can see that the ideal curve in this particular figure, you can see the dashed lines is indicating the ideal curve should be 40 mg for transport maximum of glucose. The ideal should be that till this level, this is the rise in the transport maximum, and suddenly all the carriers get filled, completely occupied, and then it becomes flattened. But in actual circumstances, this is not the case. Actually, the curve, it is rounded at this particular area. It is rounded, and this is known as splay. Okay, so this phenomenon of rounding of curve of TMG is known as splay. So what is splay? It is the rounding of the glucose reabsorption curve. And what is the reason for this splay? The reason is that all the nephrons do not have the same filtering and absorbing capacities, right? Some nephrons, they have a different type of capacity. They have less amount of carrier, so the glucose will not be absorbed from these nephrons. Some will take a large number of carriers are there, and therefore they can absorb to a greater extent. So due to varying capacity of absorption of glucose in all these nephrons, this is the ideal curve usually do not appear, and we see this type of curve in the TMG, and this is known as splay. The second reason can be that the glucose carrier does not have an infinitely high affinity for the glucose. So glucose escapes in the urine before the carrier is fully saturated, right? So there are two mechanisms by which due to which this splay appears. First of all the nephrons do not have the same level of TMG, do not have the same level of carriers. Some get saturated earlier, some get saturated late. Secondly, all the carriers do not have a high affinity for glucose. Some do not function that effectively, and the glucose is not reabsorbed from them, and they do not get fully saturated, and they do not get fully reabsorbed from them. So due to these variations, there is a splay in this curve. Now this curve, this diagram is again showing the reabsorption of water and solutes, and mainly all these are coupled to sodium reabsorption. So we have seen till now that the main substance which is causing the reabsorption of all other substances is the sodium. And why? Because the most effective pump which is functioning in the cell is sodium potassium ATPase pump, and because of its functioning, a lot of things are taking place. We have already seen that due to the entry of sodium inside the cells, there is secondary active transport of glucose or amino acids inside the cell. We have also seen that due to the entry of sodium, an antiport, sodium hydrogen antiport, is also functioning, causing the secretion of hydrogen from the cells. Then there is movement of chloride ions also along with sodium, because the negativity is also developed here, because more and more sodium is entering inside the cell. So negativity is developed this side, which pulls the chloride ions inside the cell. Water, of course, will follow all the ions and other substances, osmotically active substances. When they are reabsorbed, water will always follow. And at various places, urea is also reabsorbed at various places. So the mechanisms of coupling water, chloride, and urea reabsorption with sodium reabsorption is shown in this particular flow diagram. What we see here is, with the sodium reabsorption, there is an increase in negative potential being developed in the lumen. In the tubular lumen, increased negative potential is developed, and because of that, passive chloride reabsorption will occur. Secondly, due to sodium reabsorption, water reabsorption will also occur. And due to more and more water reabsorption, increased luminal chloride concentration will occur. Again, this leads to passive chloride reabsorption. Then on the other hand, due to water reabsorption, increased luminal urea concentration will also occur, leading to passive urea reabsorption. Okay, so these are the various mechanisms which are also linked with the reabsorption of sodium. So we have seen that due to active transport, due to secondary active transport, so many substances were being transported along with sodium. Apart from it, due to various mechanisms like increased negative potential being developed, increased amount of chloride concentration developed due to water reabsorption, passive chloride reabsorption is also taking place, and water will always follow any osmotically active substance being transported. So increased urea concentration will be there when water is being transported. So passive urea absorption will also occur. Now coming one by one to different portions of the tubule, different parts of the tubule, speaking up abundance which are there taking place in the PCT, in the different tubules, portions of the tubules. Now this is the figure of proximal tubule. We can see here the first component of the tubule after the Bowman's capsule is the PCT. So the maximum absorption, reabsorption, that is 65% of reabsorption occurs in PCT. PCT, I had already shown this cellular structure of PCT earlier, where I have shown that it has got a maximum absorptive capacity, lot of villi, increased surface area, showing lot of absorptive capacity. It is a very active cells. The cells here in PCT are very, very active. They show large amount of mitochondria. There's lot amount of activity taking place, and therefore it is responsible for maximum amount of reabsorption. So the substances which are reabsorbed in PCT are all these ions mentioned here. Of course, water will follow, glucose and amino acids. The mechanisms which we have already discussed, active transport, secondary active transport, passive diffusion, osmosis, they all will be applied here in PCT, and they will all cause the movement of these ions. This arrow is indicating reabsorption of these substances. This is from the lumen towards the interstitial tissue. These are the reabsorption of substances taking place in PCT. On the other hand, hydrogen ion, certain organic acids and bases, they get secreted in PCT. So what is happening due to reabsorption in proximal tubules? The filtrate is reduced to 1/3. The fluid which is leaving the PCT is isotonic to plasma because tonicity is continuously being maintained due to movement of water. Whatever amount of substances are reabsorbed, the water will proportionately flow and try to maintain the osmotic balance. As I told you, that water will will be reabsorbed osmotically, so it will try to maintain osmotic balance. The substances which are completely absorbed in PCT are glucose and amino acids, while sodium bicarbonate and chloride are more than 99% absorbed at the level of PCT itself. And we have already discussed that how the whole mechanism is based on the absorption of sodium. I hope the PCT portion is clear. The substances which are being absorbed in PCT, and as I told you, it is 65 to 67% of sodium being reabsorbed in PCT, and all other substances, most of them are dependent on sodium. So most of the substances, 67%, are absorbed in PCT. Then as we go to thin loop of Henle, thin limb, descending portion, ascending portion, and thick ascending portion, 25% of reabsorption will occur in thick ascending loop of Henle. Then 5% will occur in DCT, and 3% will occur in collecting ducts. We'll be going one by one to all these portions of the tubules where different reabsorptions will occur. This is the changes in concentration of different substances taking place in PCT. So this graph detects, and this line, isoosmotic line, is shown here red color. That is, this is showing red color. Dotted line is showing osmolarity. And this is showing the concentration of these substances are remaining constant. Although amount of substance is very much being decreased, sodium, so much reabsorbed, 65% is reabsorbed in PCT. But since water is also being reabsorbed along with it, the osmolarity is remaining constant. The concentration is remaining constant. Same is true for chlorides. But what you see for other substances which are maximally absorbed, like glucose and amino acids, which I told you are almost 100% reabsorbed in the PCT, their concentration goes on decreasing till the terminal portion of the PCT. Same is the case. Similar is the case for bicarbonate. You can see because it is quite a lot absorbed in PCT, so its concentration also falls. While substances like creatinine and urea, they're not reabsorbed. So you can see that concentration keeps on increasing. So what determines the concentration of a substance? The concentrations of solutes in different parts of the tubule depend on relative reabsorption of the solutes compared to water. Okay, so it is always in comparison to water. If the water is reabsorbed to a greater extent than the solute will become more concentrated in the tubule, as we have seen the case of creatinine and inulin, isn't it? The water is getting reabsorbed to a greater extent than these solutes, then their concentration keeps on increasing. On the other hand, if the water is reabsorbed to a lesser extent than the solute will become less concentrated in the tubule, as is the case of glucose and amino acids. Glucose and amino acids are completely reabsorbed in the PCT, while if we compare it with water, they are much more reabsorbed and therefore their concentration decreases. I'll show this graph again. You can see clearly that these substances, their concentration goes on decreasing because water is reabsorbed much more. These substances are reabsorbed much more than water. So their concentration falls, while these substances are not reabsorbed much, so their concentration will increase, while sodium and chloride, they follow, the water follows according to their thing, and their concentration almost remains constant and the osmolarity is maintained. Coming to the next component of the tubule, that is thin descending limb of loop of Henle. So here I'm discussing in this particular diagram the thin descending limb of loop of Henle. The descending limb is always thin actually. The ascending limb has got two components, thin and thick. So we are talking about the descending limb of loop of Henle. You can see flattened cells in this. Not much activity of these cells are observed, and it is very permeable to water. It is not permeable to solutes, but it is very permeable to water. While this figure tells us about the thick ascending loop of limb of the loop of Henle, where 25% of reabsorption is taking place. So you can see these many ions are being reabsorbed. Here also the activity of the cells increases, the cells become taller, and all these reabsorption is taking place. So 25% of the filtered load is reabsorbed in the thin ascending limb of the loop of Henle. There is reabsorption of all these ions. You can see sodium, chloride, potassium, bicarbonate, calcium, magnesium, cetition, hydrogen also, and it is not permeable to water. Okay, so this is very important difference between the descending limb and the ascending limbs of loop of Henle. The descending limb is very well permeable to water, very much permeable to water, while the ascending limb is not permeable to water. It is very much permeable to other solutes, other ions. So reabsorption, the remaining 25% of the filtered load is reabsorbed in this area. And this will be helpful. This particular concept will be helpful when I'll be telling you about the concentration of urine. In some other lecture, we'll be discussing how the urine gets concentrated, the countercurrent mechanism, etc. will be discussed later. So this particular property of the tubules will be very helpful there. So the descending limb, again, I'm telling you, the descending limb is permeable to water, while the ascending limb is permeable to solutes, not permeable to water. So, talking about the thick ascending limb of loop of Henle, the thick ascending loop of Henle, which I told you is permeable to various substances, various ions. The most important channel which is working here is this particular channel. This channel is known as sodium potassium 2 chloride channel. That means this channel transports 1 sodium, 1 potassium, and 2 chloride ions at the same time, right? So it is named as sodium potassium 2 chloride channels. This is the most important channel which is functioning at the ascending limb of loop of Henle, thick ascending limb of loop of Henle, right? So because of this particular channel, and you know that what is driving this channel also, this particular thing, this primary active transport, which is taking place at the basolateral membrane. And due to this, the sodium is driven in, and along with it, there's a channel which transport 1 potassium and 2 chloride ions along with 1 sodium ion. So these ions also move inside. Now, out of these ions, chloride will pass out from the cells passively, while some amount of potassium will pass out, and some amount of potassium will also be secreted back. Apart from it, there is antiport also working here, sodium hydrogen antiport also working here in this portion of the tubule. And some ions, other ions, they are transported through the paracellular route, because water is also diffusing through paracellular route. So many things, many substances, many ions also gets diffused along with water. Now, there's one more important thing written here, loop diuretics. We'll be discussing diuretics in detail later, but I just want to tell you diuretics are the substances which increase the amount of urine. That means they prevent the reabsorption of water at various sites of the tubules. So when they prevent reabsorption of water, more amount of urine can be formed. So here, if any substance stops this channel, blocks this channel, the solutes will not be transported inside. This is the main channel operating here. Main channel that is transporting solutes in the thick ascending limb of loop of Henle is sodium potassium 2 chloride channel. So if this is blocked, these ions will not be transported inside, and as a result, water will also not follow them. And as a result, this will lead to increased concentration of solutes in this portion of the tubule, and due to increased concentration passing on to the later portions of the tubule, water will not be transported. This will be one of the diuretics. Diuretics are substances which increase the amount of urine, increase the amount of water, and thus increase the amount of urine. So the substances which are acting here are known as loop diuretics because these are acting on the loop of Henle. We'll be studying this later on. I'll classify them later on also. Coming to distal convoluted tubule, we will study it in 2 portions, early portion of DCT and later portion of DCT. So right here is early DCT, early portion of the distal convoluted tubule. In the early portion of the DCT, we have an important channel, that is sodium and chloride co-transporter is present here, which carries sodium, and along with it carries chloride into the cells from the lumen. Okay, again, you know, the primary active transport is always acting here. So sodium is entering here the cells because of it, and chloride is also pulled along with it. So this is a co-transport. This is the main channel operating here. This also helps in a lot of transport of sodium and chloride. And if this channel is also blocked, again, the amount of concentration of these ions will increase in the tubular lumen, again leading to more amount of water and more amount of excretion of water also. So these substances which act here to block this particular channel are classified as thiazide diuretics. Okay, so these are the channels, sodium chloride channels, sodium and chloride co-transporters, which are the main channels present in the early distal portions of the tubules. Actually, early distal tubule is functionally similar to thick ascending limb of loop of Henle. It is not permeable to water, therefore it is also called the diluting segment. There occurs active reabsorption of all these ions here. And one more thing to remember, it contains macula densa. We have already studied macula densa in the previous class and how this macula densa is causing tubuloglomerular feedback, right? So depending upon how much sodium is delivered to this portion of the tubule, it gives the feedback to the afferent arterioles and accordingly cause its constriction or dilatation and accordingly alters the glomerular filtration rate. So this is also a function done by distal tubules, the macula densa cells present in the DCT. Now coming to early and late distal tubules and collecting ducts. This diagram is showing the early distal tubules, which we have already seen earlier also, how the early distal tubules are functioning. They are causing the reabsorption of about 5% of the filtered load is reabsorbed here, namely NaCl. Apart from other ions, it is not permeable to water and it is not very permeable to urea also. And the second figure is that of late distal tubule and collecting ducts, collecting tubule. That's the cortical portion. So here in this portion, that is this portion, the late distal tubule, you can see in this figure, the late distal tubule and upper portion of the collecting ducts, that is the cortical portion of the collecting ducts. It is permeable to water, but this permeability to water will depend upon the amount of ADH. I hope you remember what is ADH, antidiuretic hormone. So depending upon the ADH and the action of ADH, the permeability of water will occur in this portion. Now this portion is not very permeable to urea as well. You can see what all substances, what all ions are being absorbed from this portion. We can see here that sodium and chloride is very well absorbed here. Water is absorbed here, but that is dependent on ADH. Other ions also like bicarbonate and potassium gets reabsorbed here. But the two important ions which get secreted are hydrogen ions and potassium ions. This is also very important. Potassium ion also gets secreted here. And also one more thing which you can see here in the structure of the cells is there are two types of cells. One are principal cells. The other ones are intercalated cells. So I'm not going into detail of the structure, but these two cells have different kind of functions. One is principal cells and one is intercalated cells. So this is the function carried by the principal cells. The principal cell of the late distal tubules and cortical collecting tubules, they secrete potassium. Okay, so they are absorbing sodium and they are secreting potassium, and this is mainly under the control of aldosterone. So aldosterone is causing these changes here. So if aldosterone is obliterated, or we can say that aldosterone antagonists are given here, this particular activity will not be performed. Either we can give a sodium channel blocker, or if we obliterate the aldosterone or antagonist of aldosterone is given, then this particular transport will be affected. And as a result, this will also act as a diuretic, because this is important ion which is being reabsorbed, which will not be reabsorbed here. So this will also act as a diuretic. Right? So in the principal cells, the main change which is taking place, the main action which is occurring is the sodium-potassium exchange which is taking place. Potassium is being secreted here and usually under the influence of aldosterone. The other cells which are there in the late distal portion and collecting tubules are the intercalated cells. And these intercalated cells, you can see the activities going on in the cells. The carbon dioxide and water combine to form carbonic acid. You know all this reaction, it is taking place in various other cells of the body. Carbon dioxide enters inside, it combines with water to form carbonic acid, which then dissociates into bicarbonate ions and hydrogen ions. right? And bicarbonates here is shown to move inside, and hydrogen ions are transported outside in exchange of potassium. So here, what is acting? Hydrogen, potassium, ATPase is there, and hydrogen is being secreted. On the other hand, in type B cells of intercalated cells, it's written here type A and type B. There are two types of intercalated cells. In type A cells, they help in secretion of hydrogen ions, while type B cells help in the secretion of bicarbonate ions. Okay, so now you can see that how these cells are helpful in regulating acid-base balance. So whenever acid-base imbalance occurs in this portion of the tubules, accordingly a particular cell become activated and try to bring the pH back to normal. So they help in regulating the pH, because type A cells are having hydrogen potassium antipodes and they secrete hydrogen ions, while type B cells are secreting bicarbonate ions, and according to the pH, these cells becomes active and they accordingly work and secrete the corresponding ion and try to bring the pH to normal. So again, here we find the functional characteristics of late distal tubule and cortical collecting duct. This portion is impermeable to urea. The principal cells reabsorb sodium and secrete potassium, which is controlled by aldosterone. The intercalated cells secrete hydrogen ion by an active mechanism. Therefore, they help in acid-base regulation of the body fluids. And the permeability of water in this particular cells is controlled by ADH. So only with the action of ADH, the permeability of water can be increased or decreased. Coming to the medullary portion of the collecting duct. The portion of the collecting duct shown here in dark color is the medullary portion of the collecting duct, that is, which is extending towards the medulla. Medullary portion of the collecting duct is again permeable to the common ions, which are permeable most of the places, sodium, chloride, bicarbonate. But the most important ion which is reabsorbed in this portion is urea. Urea was not being reabsorbed in other cortical portions. So urea is the main ion which is reabsorbed in the medullary portion of the collecting ducts. Again, the reabsorption of water is dependent on ADH. We also find some hydrogen and bicarbonate, hydrogen being absorbed and bicarbonate being absorbed and hydrogen being secreted in this particular portion of the collecting ducts. So medullary collecting ducts are mainly responsible for reabsorption of urea and water in the presence of ADH. So permeability of water in this portion is controlled by ADH. It is permeable to urea. It secretes hydrogen by an active mechanism and therefore help in acid-base regulation of body fluids. So here you may see this complicated graph, but what is it representing? It is just representing the concentrations of various substances in the renal tubules. On the x-axis, you can see the proximal tubule. This portion is showing the proximal tubule, loop of Henle, distal tubule, and collecting tubule. So how the concentration of substances vary as the fluid passes through the nephrons, passing from all these neurons. So what we see here is, just a second. Okay. So as we have already discussed, glucose, amino acids, etc. Glucose, amino acids are completely absorbed in the proximal tubules. So their concentration goes on decreasing. It's almost lost after, not under normal conditions. It's completely absorbed in the proximal tubule. So we do not find them in loop of Henle and other portions. Even bicarbonate goes on decreasing. Sodium and chloride, I have already told you, dependent on water. So it is in relation to water, you can see their fluctuations. While these substances, creatinine, inulin, urea, they keeps on increasing once the fluid passes from PCT to loop of Henle to DCT and the collecting tubules. So this reabsorption is regulated by various mechanisms. So we have already discussed various mechanisms like the ones which are there. Okay, I'll tell you these hormonal and other mechanisms later on in my next class. Only one mechanism I want to explain today, and that is glomerular tubular balance. That means the tubular load is directly proportional to reabsorption. The reabsorption is directly proportional to the tubular load. That means a form of auto-regulation is there in the tubules also, known as glomerular-tubular balance. That is the intrinsic ability of the tubules to increase their absorption rate in response to increased tubular load. So if there is more tubular load, more amount of, they will increase their capacity to absorb. If GFR increases from 125 ml per minute to 150 ml per minute, the absolute rate of proximal tubular reabsorption will also increase from 81 ml, which is 65%, to 97.5 ml per minute, which is again 65%. So whatever amount is filtered out, 65% of this will be reabsorbed. This is glomerular tubular balance. So tubules try to maintain and try to increase the reabsorption, and this is by a fraction which remains same. And this helps in preventing the overload of DCT when GFR increases. So this is the main function, and this is also one of the components of autoregulation. We have already studied autoregulation as far as tubulo-glomerular feedback was concerned earlier. Now this is the autoregulation which is taking place in tubules known as glomerular-tubular balance. That means tubules are absorbing a fixed fraction of whatever load it is receiving. Now whenever we have till now studied how the substances are reabsorbed inside the cells and they are transported to the interstitial tissue, finally they will be transported to the peritubular capillaries by means of hydrostatic and colloid osmotic forces. Okay, so whatever it is, mainly all these substances, they follow the bulk flow and they are transported into the capillaries, because so much solutes and so much fluid gets accumulated here that due to bulk flow, they all enter the peritubular capillaries, but they follow the principles of hydrostatic and colloid osmotic forces. Now, one of the doubt which was there in the last class, few people had asked that why protein ingestion, or we can say that glucose levels, increases the GFR. Whenever there is increased protein ingestion, there is increased amount, because protein will degrade into amino acids. It will be digested to amino acids. There will be more amount of amino acids. So more proximal tubular amino acid reabsorption will be there, because infiltrate, more amino acids are there. So more will be reabsorbed in the proximal tubules. So more proximal tubular NaCl reabsorption will occur. So, and how does this occur? If you remember this particular pathway, because glucose and proteins and amino acids, they are reabsorbed by secondary active transport. So whenever more amount of amino acids or more amount of glucose is reabsorbed, more amount of sodium also gets reabsorbed. When this is reabsorbed, less amount of sodium will reach the macula densa cells. And when less amount of sodium is reaching the macula densa cells, it will decrease the resistance of afferent arterioles, causing increase in GFR. I hope that doubt is clear. Same is true for glucose. Whenever there is increased glucose levels and more amount of glucose is being filtered, more amount is reabsorbed in these cells, less amount, and as a result, more amount of sodium will also accompany it, less will reach the macula densa, and it will try to increase the GFR. That's all. And that's all for today's lecture. Now, one important announcement I have to make. We took your test yesterday. Some of you, I agree, were quite serious and did it seriously, study and do it seriously. But some of the students, it seems, are not at all serious. They are taking all these things as if it's a joke. Whatever the teachers are putting a lot of effort to teach them by this particular way, to prepare for the exams and conduct their exams, and some people are just taking it jokingly. This is not at all acceptable. And we know who all these people are. We always come to know all about this. So please be aware and please be very serious from next time. No more false submissions. You have submitted many responses, which we have seen that writing whatever name you want to, you write. You all are getting 0. Just 0 if you do this from this exam also. And these all marks are your internal assessment. Don't think this is a joke and there's a play going on here. This is all added to your internal assessment. Okay, so this is your all marks are being added, and you know that this is in the new curriculum. You will not be allowed to sit in the exam if you are not scoring 50%. So please keep a check on the marks. We'll be giving you these marks also. The previous test marks are already declared to you. These are also we are giving very soon. And you see, check yourself if you are not getting 50%. This is an alarming signal. Start reading now. You have enough time. Don't waste your time. Ask whatever doubts you have, but don't waste your time, please. And this is the last warning. No more nonsense from the next test. Thank you.